Okay, this is the last section of chapter 24. We just finished up talking about how checkpoints can either pause the cell cycle to allow for DNA repair, or they can trigger this process called apoptosis. So in the event that a cell cannot repair the damage to its DNA, and following the arrest of the cell cycle by a checkpoint, the DNA damaged cell has to be eliminated. And so in many cases, the process has to be conducted in a way that the damaging cells in the tissue aren't also damaged. And so multicellular organisms accomplish this through a process called programmed cell death, also known as apoptosis. So this isn't unique to just DNA repair mechanisms that fail triggering the process. It's also used by numerous other physiological processes, including those that work to eliminate infected or otherwise diseased cells in bodies. There's another type of cell death which is less specific, called necrosis, which sometimes occurs following tissue injury, and that involves swelling and rupture of injured cells, whereas apoptosis is more of a specific series of events that leads to dismantling of the cell and different cellular structures, so it's much more organized than is necrosis. So in terms of the process, looking at the process overall, first thing that happens is that the cell's DNA segregates near the periphery of the nucleus, and the cytoplasmic volume begins to decrease, so the cell begins to shrink up and detach from the substrate that it's growing on. You can see the cells here are starting to shrink up and round up. The cell then starts to produce small cytoplasmic extensions. You can see these they look like phytopody, but they're not. And the nucleus then starts to fragment and break down. And the DNA is cleaved by a apoptosis-specific endonuclease, or DNase, and the cell is then systematically dismantled into small pieces, these small pieces of the cell up here, which are called apoptotic bodies. And then one of the last things that happens is that a phospholipid translocator that we talked about before, actually one of these flipases, is inactivated, and that leads to the accumulation of phosphatidylserine in the outer leaflet of the plasma membrane, and that serves as a signal for phagocytic cells, macrophages, and that kind of thing to come and engulf the apoptotic body, and so that's a specific function of phagocytosis. In terms of the actual mechanisms look kind of complicated, but apoptosis proceeds generally through the activation of a series of enzymes, a series of proteases called caspases. And caspases are produced as inactive precursors. Now, you don't want to be expressing these caspases in a form which they can automatically just start breaking the cell down. And so they're expressed in a form called a procaspase, and then they have to be cleaved to create active enzymes. And we've talked about that kind of process before, so we won't get into it again. Now, looking at the diagram here, there are kind of generally two main routes. One at the top here, where activation of the apoptotic pathway can occur directly. For example, if this cell was infected by a virus, then cytotoxic T cells, these cytotoxic T lymphocytes, can recognize this infected cell, and using death receptors, the activation of death receptors can trigger one pathway up here, and so that's triggered in response to the reception of death signals. And the other general mechanism involves the withdrawal of survival factors, and so a survival factor would switch off the apoptotic pathway, and so withdrawing the survival factor allows the default apoptotic pathway to be invoked. So those are the two general mechanisms. If we take a look at things in a bit more detail here, here's a cytotoxic T cell, and on its surface is a ligand for a death receptor. And so if this cell here has recognized this cell as being infected by a virus, for example, then the ligand engages the death receptor, and that causes the aggregation of some of these procaspases, notably procaspase 8, and they become, they're activated, and they are then able to activate a series of a group of proteins called the executioner procaspases, for example, procaspase 3. And so the initiator caspases cleave the executioner caspases, and then the executioner caspases are involved in the proteolytic breakdown of a lot of the molecules in the cells, and so that triggers apoptosis. The other mechanism we've got going on down here is the removal of survival factors. And some of the things that happen here are similar to what happens up here. So let's look at the bottom pathway. So if you withdraw a survival factor, that allows the accumulation of death-promoting proteins to accumulate. So normally this receptor with a ligand bound will actually lead to will suppress the death-promoting proteins. And so if you take away the survival factor, the survival signal, these proteins can accumulate. That ultimately leads to the release of a protein called cytochrome C from the mitochondria, and cytochrome C binds with a protein called APAF1 and procaspase 9 to form an initiated caspase called the apoptosome, and that can activate execution of caspases, and they can then go and trigger apoptosis. So there's a lot of similarities between these two pathways. Just be aware of the fact that there are initiated caspases and execution of caspases, and then these two general pathways. You don't need to remember all these different kinds of molecules as well. Lastly, notably at the bottom here, you can see that DNA damage can activate p53 through the mechanism we talked about just now. So DNA damage is recognized by ATM, activates CHEK1, and then CHEK1 can phosphorylate p53. One of the things that p53 can do is trigger apoptosis through this pathway. So that's it for apoptosis, and that's the end of chapter 4.